Your morning breakfast is a sequence of chemical interactions. Between the bread, the spread, and the morning coffee, a typical consumer encounters a dozen symptomatic emulsifiers, preservatives, and colorants before leaving the house. Under EU law, every one of those substances is authorized as safe. The European Food Safety Authority has reviewed the toxicological data for each chemical and set strict limits on their use. But that safety guarantee is built on a specific condition. It assumes you are consuming these chemicals one by one in total isolation from each other. The biological reality is different. In the supermarket aisle, a single processed product frequently contains 3 to 10 different additives, all entering your system at the same moment. While the EU's individual approvals are legally compliant, they don't account for the reality of the chemical cocktail we consume every single day. Current safety testing is conducted in a vacuum. Regulators evaluate a single substance over a short duration using healthy animal models in highly controlled conditions. This laboratory setting doesn't simulate a human lifespan. We are exposed to these chemicals starting in childhood and continuing uninterrupted for decades. This is a mapping of the regulatory blind spot. While a single additive is proven safe in a controlled environment, there is no mandatory testing for how these specific co-occurring chemicals interact once they mix inside the human body. Relying on isolated short-term data to predict the safety of a lifelong chemical cocktail creates a systemic gap in our understanding of public health. Consider emulsifiers, used to maintain texture in everything from ice cream to bread. In standard toxicity tests, they pass easily because they don't cause immediate acute harm. However, independent studies identify a secondary effect, microbiome dysregulation. Daily exposure to certain emulsifiers alters gut bacteria, degrading the intestinal barrier and increasing permeability. This degradation triggers a low-grade chronic inflammatory response. It disrupts the metabolic markers that keep our systems in balance, creating what researchers call metabolic dysregulation. These barrier function biomarkers are early warning signs. They transform a substance that is safe on paper into a biological precursor for metabolic syndrome and inflammatory bowel disease. Regulators apply the same math to nitrites in processed meat, setting an acceptable daily intake based on the isolated chemical. That math ignores the food matrix. During industrial processing and human digestion, nitrites react with the proteins in the meat to form entirely new compounds. This tracks the chemical transformation. Nitrites interact with the meat matrix, producing nitrosamines, compounds recognized as a primary contributor to colorectal cancer. The interaction is so well documented that the International Agency for Research on Cancer classified processed meat as a Group 1 carcinogen. By focusing on the additive in isolation and ignoring its reaction within the food matrix, the regulatory model miscalculates the true risk to the consumer. The EU framework relies on a system of massive technical dossiers. To authorize an additive, companies must submit extensive toxicological reports to prove their product won't cause harm. But those primary dossiers originate from the companies themselves. The system is built on data generated by the applicants seeking approval. There are few safeguards for systemic independence. There is no dedicated EU fund to pay for independent replication studies, and proprietary data often prevents third-party scientists from conducting their own reanalysis. We don't need to allege misconduct to see the risk. A safety assessment process built on proprietary, applicant-generated data is structurally compromised. The problem isn't a lack of rules, but an outdated model. Regulators are using a 20th century testing system to govern a highly complex 21st century diet. Reform requires 3 structural changes: mandatory mixture exposure triggers, whole diet cumulative modeling, and an open platform for reporting real-world health signals. The metabolic dysregulation and barrier function biomarkers we see in the lab are the same markers linked to the rise of obesity and type 2 diabetes. These are the signals the current framework is missing. A meal is not a static list of isolated ingredients. It is a lifelong, cumulative chemical reaction. Until the EU updates its framework to test food the way humans actually consume it, the consumer remains the subject of the ultimate cumulative trial.